Hey guys, welcome back to One Stop Biology. So in today's video, we are going to cover some cytoskeleton. We are going to cover some locomotory organelles that we have like cilia and flagella, and then we'll cover centrosome and centrioles. So guys, before we start off with the video, please do not forget to give your feedback on the video so that I can improvise, and please do not forget to like it and do share it with your friends if you understand this video completely and if you like it. So, guys, let's start with the cytoskeleton. Now, cytoskeleton, as it refers from the word itself, that the skeleton of the cell, right? Cyto means cell and skeleton means the framework of the cell. So what happens is, guys, that there is an elaborate network of what? Network of filamentous proteinaceous structure. So structure made up of filamentous protein, right? In the cytoplasm. So basically, we have an elaborate network of filamentous proteinaceous structures which is present in the cytoplasm, and these structures collectively are known as cytoskeleton. These structures are collectively known as cytoskeleton. So we have network of filamentous filaments like protein, so proteinaceous structure, and this is present in cytoplasm of the cell. And these structures together are known as cytoskeleton, right? So cytoskeleton, what is the function of cytoskeleton? Cytoskeleton in a cell are involved in many functions. So basically, the functions of cytoskeleton are first is to provide mechanical support to the cell. So they provide mechanical support. They give, in many cases, motility to the cell. They maintain the shape of the cell. So they help in maintaining the shape of the cell. So these are exoskeleton, these cytoskeleton basically provide mechanical support. This in sometimes give motility, and they also help in maintaining the shape of the cell, right? So these are nothing but the concept of and the functioning of cytoskeleton. Now, what is cilia and flagella then? So cilia and flagella are some outgrowths. So they are what? Hair-like, hair-like outgrowth of what? Of cell membrane. So in many cases, what happens is you'll find hair-like outgrowths of cell membrane. Now, what is the difference between cilia and flagella? Cilia is smaller, right? So these are small structures, right? So these are small structures which work like oars, and it works like oars. So guys, do you know what oar means? What is an oar? Oar is, the object which you use to row your boat. So you use a wooden stick, right? Wooden stick which is flattened at one end, which you dip in water and you row your boat, right? So it is something like an oar, and it is a small structure. Basically, it works like how the oar of a boat works, and it therefore helps in movement either of the cell or of the surrounding fluid, right? Now, what happens in flagella? Flagella is comparatively longer, right? So it is longer, And again, it helps in cell movement, right? So you must have seen in different structures of bacteria. So the prokaryotic bacterial structure have flagella, right? But these are structurally very different from the eukaryotic flagella. Remember that, right? Now, if you study the cilia or flagella in under a electron microscope, you'll see that they are covered with plasma membrane. Basically, they're made up of the cell membrane. They're outgrowth of cell membrane and they are usually covered with plasma membrane. And the core of cilia and flagella are known as axoneme, right? So this axoneme possess a number of microtubules which are running parallel to the long axis. So there is an axis and you have various microtubules which are running through to that parallel axis, right? So this axoneme usually have 9 pairs of doublets, double, right? So in total 18. So they have 9 pairs of doublets of radially arranged peripheral microtubules and then a pair of centrally located microtubules. I'll show you the same in this diagram as well. This is the internal structure of a cilia and flagella. So what I said was that they have 9 pairs, 1, 2, 3, 4, 5, 6, 7, 8, 9. So 9 pairs of doublets, right? 9 pair of doublets of radially arranged peripheral so these are peripheral microtubules, right? And then a pair here, right? So a pair of centrally located in the center, centrally located microtubules, right? This arrangement of axonemal microtubules are referred to as 9 plus 2 array. This is 9 plus 2 array, right? Now the central tubules are connected by a bridge, right? In between. This is the bridge, right? And these are enclosed in a central sheath. So this sheath that you see on both sides, this is central sheath, right? Now the central sheath is connected to one of the tubules of each peripheral doublets by a radial spoke. So here you see these spokes, right? So see, here it is connected by these spokes. These are radial spoke. So the central sheath is connected to one of the tubules of each peripheral doublets by a radial spoke. Now there are 9 radial spoke. For each pair, there are 9 radial spoke, right? Now the peripheral doublets are also interconnected by linkers. So there are linkers which connect the peripheral doublets, right? And both the cilia and flagellum emerge from centriole-like structure which are known as basal bodies. Basal bodies. So both emerge from basal bodies. So see again, this is the central microtubule, right? These are the peripheral microtubules and these are doublets. Here you have the bridge, inter doublet bridge, and inside you have the plasma membrane. So this is the structure of cilia or flagella. Now the third organelle that we are going to study is centrosome and centrioles. It is centrosome and centrioles, guys. Now, what is a centrosome and what are centrioles? Basically, let's see that. So, centrosome is an organelle which has two cylindrical structures, and these cylindrical structures are known as centrioles, right? Now, these are surrounded by what? They are surrounded by amorphous pericentriolar materials, right? So there is an organelle which is centrosome. Inside that you have two cylindrical structures. These cylindrical structures are known as centrioles. These centrioles are surrounded by an amorphous pericentriolar material, and both the centrioles in the centrosome, they lie perpendicular to each other, right? So it has an organization like a cartwheel, right? They are perpendicular to each other, and they have an amorphous pericentriolar material around them, surrounding them, right? So basically, what they are made up of, they are made up of nine evenly spaced peripheral fibrils, and centrioles are made up of tubulin protein. They are made up of tubulin protein, and they have nine evenly spaced fibrils, peripheral fibrils, which are made up of tubulin protein, right? Now, each of the peripheral fibril is a triplet. Remember that they are made up of what? Peripheral fibril, right? Each of these fibril is a triplet, is a triplet, and the adjacent triplets are linked, right? So the central part of the proximal region of centriole again is proteinaceous, and it is known as hub. The central part of you know centriole is known as hub. It is made up of protein. Now this is connected with the tubules of the peripheral triplets by radial spokes. So there are radial spokes through which it is connected, right? And again, it is made up of proteins. So you can see that the entire structure is made up of proteins, right? Now, the centrioles basically form the basal body of cilia and flagella, right? So we ended cilia and flagella saying that they are attached to the basal body. Now, these centrioles form the basal body of the cilia and flagella, and also spindle fibers that give rise to spindle apparatus, which is used during cell division. So we are going to study cell division as well, where you'll study spindle fibers, where you'll study spindle apparatus. So these centrosome, these centrioles are basically, they form the basal body for cilia and flagella, and also spindle fibers, right? So centrioles form basal body of cilia, flagella, and spindle apparatus, right? Basically, there is a centrosome which has two centrioles, and they are made up of protein. They are made up of peripheral fibrils, which is made up of protein. They have a center, which is hub, and then they have a radial spoke, right? They have tubes inside, and they form the basal body. So, guys, with this, we have finished the next three organelles, and in the next video, we are going to start off with nucleus. Now, nucleus majorly will be the last, topic of this chapter, and then we'll start off with the next chapter of cell biology, which is biomolecules. Again, very important. So, I hope you understood the basic structure. See, just remember that these are just the basics, and we are right now just covering the NCERT. So we are going, as per the NCERT syllabus. Some of you might feel that, this information is incomplete because we are not going in depth as per the other books. Once we start off the other books as well, we'll go in depth. This is what needs to be covered as your basic if we are new to these topics, right? So basically, this is part of the NCERT series itself. So I hope you understood all of these organelles completely. Please do like the video and share it with your friends if you did. And if you have any doubt, please do not hesitate in asking your doubts. You can either mention it in the comment of the video, or you can also ask your doubts through WhatsApp. I have shared the WhatsApp number on in description of the video. But please do give me feedback if you have any. I would highly appreciate it, and I would be waiting for your feedback, guys. Thank you for watching this video. See you in the next video. Bye bye.